Alright folks, this week in lab we looked at vascular tissue of a few different plant groups. And so I want to go over those slides real quick. Hopefully everybody saw the correct things on them. I know it can be a little tough to grasp, and this is stuff you're covering in lecture 2, so hopefully get some nice synergy here. And it will help you succeed in both the lab practical and the plant test coming up very, very soon. So without further ado, let's get into it. Miss if we didn't start off with ferns. Ferns. Here's an example of a fern rhizome. There's a lot of things happening here. It looks kind of scary. A lot of creepy circles, a lot of weird things happening. What even is life? But nevertheless, this is what we're looking for. This is the vascular tissue of ferns. And you also notice there's another on the bottom. I did not highlight that one. Could you see how poorly drawn this one line was? I didn't feel like trying to draw another poor line. I thought that wouldn't do anybody any favors, so I didn't do it. But that's it. And so what you want to be able to do is note how this looks and how it's going to compare to what we're about to see in the gymnosperms and angiosperms. And in short, it's very, very different. And so we will see that soon. Closer at our fern, you can see the very clear large cells and small cells around there. And so this is our phloem on the outside, those little cells. They look green here, but that is them. And then the xylem along the inside. So the xylem is the tissue that conducts water. The phloem is a tissue that conducts the food and other things that the plant will use. And so both are considered part of vascular tissues. And note, ferns, gymnosperms, and angiosperms have this. Bryophytes do not. So this is the groups that have that, and they have these cells. And of course, this is found in the sporophyte stage, which is the diploid stage of these individuals. So they've got it. It's there. Now let's hit the gymnosperms up and see how it compares. And it's a little more complex, but it makes it also pretty fascinating. So here is our slide. Hopefully you saw something pretty similar to this. And I remember talking to some, and it was some initial confusion of what exactly am I looking at here? Because you know what? I kind of get it. If you look at it though, it looks kind of cool, like a nice little painting, of some kind. I don't think it's that bad. I don't know if I'd hang it up and put it on my wall, but I would not hang it up and put it on my wall. So maybe the bathroom or something. Hey look, this is a cool plant tissue. And now that I'm looking at it, it does kind of look like a weird eye. We have this big center point with like the colorful part around it. I don't know if you can see my mouse, but a colored part. So it's like a plant eye staring at you. It's a tad unnerving. So let's re-nerve this sucker. And here is the xylem. And so you can clearly see there's a lot of tiny circles in there. It's a purplish looking part. That is the xylem. The phloem is just outside of that, and it's smaller green circles. And so this is a pine slide, and so pines can experience secondary growth. And so they can grow and get wider. And so normally with that, the xylem sticks around while those cells continue dividing from the vascular cambium, which isn't really shown here, but it's there. It's dividing the xylem and the phloem. And so as it divides, it pushes out more xylem, expanding the width of the specimen. But the phloem will get kind of crushed along the outside, so it's usually a little smaller and flakes off and goes away. So that's kind of what's happening here. Phloem usually not going to be as big of a layer as the xylem, and you can clearly see those two. But what if we get closer? Technology allows us to enhance the image. Here we go. Here's the zoomed in version, and this does give us a little more detail to what's happening, and you could see this or the zoomed out version on the test to be prepared for both. You should be able to recognize though it's cell you're looking at, whether it's xylem or phloem, what the function of it is, so function of xylem, function of phloem, based off identification, and what phylum is it in. And then from the phylum, you could have other questions that would infer knowledge from that phylum. So something maybe like, was this, could this produce seeds? Does this have vascular tissue? Which obviously it does, but that'd be kind of a fun one to throw out there. But things like that. Does it produce fruits, produce flowers, things of that nature? So just keep all that stuff in mind. It's what you could maybe see on the practical, so keep that mind open. But let's examine this closer, and here we have our xylem. And we're zoomed in, so it's a much larger territory, and you can see all of that. And you can also note the small little holes, which represent tracheids, which are individual little cells that are responsible for transporting water in plants. So one of the cells that can do that, and there's a ton of these things. And if you note, they're all pretty much the same size in this pine. Now, there are some gymnosperms that can have some vessel elements, so they can have some larger cells that we'll see in a minute. But for our purposes, we don't have a slide of that we're going to show you. You're going to see one that looks like this for gymnosperm. And that's one of the easiest ways to tell it apart, is it has relatively same size circles called tracheids. And that makes it a whole lot easier to identify for our case. Now, I know what you're thinking. What about the phloem? The phloem. And that's out here. Tiny little squished green little circles. That is, of course, our phloem. So, looking back, we have our xylem, tracheids, phloem. That's it for gymnosperm. But now it's time to talk about angiosperms. Yeah, the alphas out there, the plant community that have taken over and ruling the area. Let's see what theirs looks like. Boom, we got it. I like the way this one looks because that initial image in the center kind of reminds me of like a cool looking sea star. That's pretty neat. I guess it's kind of a little teaser, if you will, an Easter egg coming up of what we'll talk about in the second half of the semester. But as it is, on first glance, it looks similar, but pretty key differences to the pine. And hopefully you're starting to recognize some of those as you compare them. And so this is our xylem. There's actually some unique things going on with the xylem we can actually able to see in this one compared to the pine, and that is the presence of different layers of growth. And so when plants, when it comes time to grow, there's early growth and late growth, representing a growing season, and also rings can form, allowing you to age trees potentially in the right environments. As long as conditions are good and they're growing in the right area, you can see these sorts of things. In this case, we can get a hint of it. And there is our first year of growth. And so that there will be the early xylem tissue. And you can clearly see large circles. We'll talk about what those are named in a second. And then it kind of leads into smaller circles. And then there's a second year of growth, a second little row shown, where those big circles come back. And so that's how you can kind of tell the difference between early versus late. Early will have more of those larger circles present, while late will have more of the small circles, which are our good friends, the tracheids. And then when you have all those big circles meet the previous year's small circles, that's generally the clue that you're hitting in the next growth year. And so the second year is newer cells, while the first year are older cells. And as the plant continues to divide and grow and widen, you'll have more and more of those rows that push themselves out. And it's the outermost layers that will be the most active when it comes to actual water transport. Then we got our phloem up here. Again, tiny little sliver piece. That's the phloem. It's very delicate. You could probably stretch that out a bit with all that green, but I just want to hit that area. That's the phloem. And let's go zoom closer. Here we go. So this is a zoomed in version of what we were just looking at a second ago. On the left side of your screen is the more interior part of the plant. To the right side is the more exterior. And if we look here, we can see our clear rows of xylem. So we have our first year of growth and our second year of growth. And again, you can see those tiny little cells and where they meet up with the larger cells. And so that is where you can easily see that difference there. And then you can see that happening. And then we got our phloem on the outside. So nice little phloem, greenish in this case, nested right up against the xylem. And then going into the cell area, we can see the actual tracheids, which are again similar to what pines have. Tracheids are found throughout vascular plants. But then you have vessel elements, which are not exclusive to angiosperms, but they're very often found in angiosperms. Angiosperms have a ton of these suckers. And so these are useful in transporting water in larger amounts. So their tracheids and vessel elements do the same thing of water transport, just vessel elements are bigger. And so it's a bigger pipe, and they can transport more. And in our case, for our slides, the angiosperms have them, the gymnosperm doesn't. And that fern kind of had some big ones. So we can show that vessel elements are not, necessarily a homologous, unique trait to the angiosperms only. It popped up several different times. It's a convergent evolution amongst plants. But so that's our years of growth. Hopefully it can help you recognize what you're looking at with that vascular tissue. There's also one other thing we looked at I want to touch upon, and that is the stomata. And so the stomata connects with the vascular tissue in a way, in that the vascular tissue carries water through the plant. And then one of the main drivers of water movement in the plant is the stomata, because of something called transpiration, which we probably talked about at this point. Here's the stomata we looked at. I don't know why this is so dark. It didn't look this way when I took the picture. But nevertheless, I guess I want to turn the lights out. And if we zoom in, this is the stomata. The area that circle inside of there. And so it kind of looks like a mouth to me. This one in particular looks like an eye because of that center spot. It's just so it's not like a fully clean section, so it's just some remnants built on top of the specimen that I peeled this off of and took it. That's why it kind of looks that way. That's not really typical. But what is typical are these cells you can see. And so it can be closed mouth or an open mouth, depending on if the stomata was open or closed. Their function is to, I don't know if you can hear that, my Alexa is telling me to do something. Anyway, I'll sort that in a second. But yeah, so these things will open and close to allow gas exchange. Alexa, stop. I hushed that up. So anyway, those cells they're highlighting are guard cells, and they will actually allow the stomata to open or close. They facilitate the opening or closing, which allows gas exchange, which allows the plant to do photosynthesis with the intake of carbon dioxide, and then get rid of the oxygen made as a result of photosynthesis. And when they open or close, plants lose water, and that's transpiration. And that is the size of the vascular tissue. Thank you for coming on this ride. Hopefully it helps. Good luck. See you later. Peace out.